with our introduction to the next and this time in a beautiful month of May, Cardiovascular Research Discoveries seminar. As you know, Cardiovascular Research Discoveries are a series of webinars co-organized by Cardiovascular Research and the ESC Council on Basic and Cardiovascular Science, chaired by Christian Weber, who co-chairs these seminars with me. And on behalf of myself and Christian, I have particular honor to welcome today our speakers, who will be Professor Stefanie Dimmeler and Dr. Donato Santovito. Today's seminar is focused on cellular crosstalk in cardiovascular disease, trying to look at the use of novel technologies and discoveries and approaches to address key questions in cardiovascular disease from the point of view of various processes that concern different cells and cell types in cardiovascular disease. Without not to delay, I think I have an honor to introduce Stefanie as the first speaker. I don't think she needs much introduction. Everybody knows she's probably one of the most recognized German and international scientists working on the problems of regeneration and cardiovascular biology of endothelial cells, and has made huge contributions to the field throughout recent years. It's actually, I was thinking which contribution to highlight, but it's difficult to really decide which one is the most important. That would be for her to decide. She's leading Cardiopulmonary Institute in the University of Frankfurt and is a director of Institute of Cardiovascular Regeneration, also in Frankfurt. Stefanie, welcome, and thank you very much for agreeing to speak to our audience today. And as always, I would like to emphasize that we ask you to ask questions through the chat and question and answer section, and we will forward these questions and ask them in a form of discussion after both presentations that will take place today. Thank you very much. Stefanie, please.
Thank you, Tom, for the nice introduction, and also thanks to you and Christian for giving me the chance to discuss with some of our new data on cellular crosstalks in cardiovascular disease, and particularly in the heart. I also hope to share with you some unpublished data, and I hope that maybe just respect this. And I don't want to stop the interaction in science because of this virtual format. So I trust that everybody of you are also scientists and know that these are preliminary data. So don't spread them around, but just use them for gaining increased knowledge and for interaction discussion. I will focus on the heart, as I said, on the cellular crosstalks in the heart. I think in the past, the heart has been mainly viewed from the cardiomyocyte perspective, but in the recent year, there was increasing evidence, and it's now very clear and well studied, that there are many cells which contribute to heart function, and the maintenance of the crosstalk in the heart keeps the heart in the healthy homeostasis state, or is also required in response to adaptation. You just see here the different cell types involved, extracellular matrix, paracrine factors, all of which are very important. This is also important in the heart disease settings. This is a recent review article that we were publishing in Cardiovascular Research, highlighting that not only cardiomyocytes adapt to different disease states, but also the vasculature, particularly endothelium, which is reduced, or even there's a rarefaction of capillaries in maladaptive hypertrophy. There's an inflammation ongoing. You have fibrosis. Also, there is different contributions on inflammatory cells in different states of heart failure, like ischemic heart failure, also HFpEF. And this is very important because this may be also targeted and may be required to really keep the heart healthy or make it healthy again. Also, injury response is important, and their aging plays a major role. Depending on the age of mammalians, there is a strong difference in response to injury. For example, in the postnatal stage, there is ongoing regeneration, while in adults, you can see here scarring of the apex in a mouse heart, or in the human heart, you see here this large scar. This is even aggravated in aging, where we have also baseline fibrosis and also a worse recovery after ischemia. As depicted in this cartoon, you see also that there are many different cells contributing to the injury response. I mentioned, of course, cardiomyocytes, which may be the most traumatic difference, are shown here in the postnatal stage, where they can proliferate while they can't, or only very limited, in adult and the aging states, where they additionally gain multinucleated. But also you see the vasculature adapts in postnatal, adult, or aging states, as does inflammation and fibrosis. So this should tell you that different cells are very important in the context of cardiac biology and also injury responses. And today I will, in the first part of our presentation, focus on the question how aging does affect the heart and particular cellular crosstalks in the aging heart. We were inspired by, of course, knowing that humans, if you get older, you can't hear well, you can't smell well, you can't see well. And we were hypothesizing that maybe cellular communication in the heart may be a bit resembling what we have in the global system, in the global aging status. And we were questioning how cell communication does occur in the aging versus the young heart. It's note that aging affects the heart. So even the absence of injury like infarction, you see here that the cardiac myocytes are hypertrophied. There's oxidative stress ongoing. We have senescence and hypertrophy, as I mentioned, some apoptosis and fibrosis, but the vessel function is not that well understood, and also endothelial microcirculatory dysfunction occurs in aging. To dissect different cells, we started already some years ago in these using single nuclei sequencing. At that time, we developed single nuclei sequencing because cardiomyocytes, as you know, are very large and can't be easily used for droplet sequencing. So we isolated the nuclei of either young or old mice and then performed single cell analysis of the total heart cells. So not purifying or isolating some cells, but just taking the whole heart. And for those of you who are not yet exposed to single cell sequencing, this is what it's doing. So in contrast to previous studies where we use bulk RNA sequencing or using isolated cells, where you always have a mixture in gene expression in the isolated cell type studied, single cell sequencing allowed you to study the transcriptome of each different cell, and thereby you can cluster to different cell types and then look into the individual cell transcriptome and understand the differences also within one cell type and one population. My student always said this is like a fruit salad in comparison to a smoothie, so much better technology to pick the fruits you like or don't like, maybe also see the ones which are bad and in, for example, in disease states. This is just a quality control of the study. You see here roughly 30,000 nuclei from young and old hearts. This on the left side, you see here the clustering of the different cells. As said, for those of you who are aware of this technology, it will be very easy to see. But for those of you who are not, each dot represents a cell. And according to the similarity of the transcriptome, cells cluster together. You can see here, for example, cardiomyocytes, you see the endothelial cell clusters, you see fibroblast clusters, immune cells, and so on. And this just shows you that the mouse hearts we used, the free mice, were quite okay. So we had a population of all different clusters. And with this, we asked the first question, namely, what is affected by age? So what cell types are responding to aging in the heart? And this was done by analyzing the differentially expressed genes, so the significantly regulated genes. And on the upper circle, you see here the upregulated genes, and the lower circle, you see here the downregulated genes. And the colors depict the different cell types. And you can easily see that the majority of genes regulated are in fibroblasts. This is fibroblast population A. This is fibroblast population B. So almost half of the regulated genes were from these cells. Second were cardiomyocytes, and then endothelial cells, which also showed a significant change in gene expression. A second question which we answered was, how did the cells respond to aging? You know that senescence can occur in aging, which would maybe predict that cells have a similar response to aging, but this was not the case. This shows you the significantly regulated genes, and here you see only 11 genes were regulated in all different cell types, while the others were quite cell type specific. So this brought us to the conclusion that, first of all, fibroblasts are the most affected cell in the aging heart, and that genes are regulated in the cell type specific manner, suggesting that the single cell technology is also important because Only then you can depict the changes in the aging heart, which are quite cell type specific. Because this was the most affected cell population. And this is now a sub-analysis of the fibroblast clusters. And here we use the so-called trajectory analysis, meaning we ordered the cells according to cell states. You see here in red the young, the old cell nuclei, in blue the young. And you can also appreciate that already here there is an ordering of nuclei. So in the blue ones here, these states that are overrepresented by young heart states. And here you have the age-associated states 1, 2, 3. Then we looked into the gene expression patterns in these states and found, surprisingly to me at least, that angiogenesis was very high in the aging cluster 1. Also inflammation, this was not surprising, but this was augmented very strongly in both clusters. And we also found osteoblast differentiation in the fibroblasts of the aging heart, which was not seen in the young heart cells, where we mainly saw extracellular matrix structure and organization. In summary, this showed us, the short summary showed us that there is changes in angiogenesis-related genes in the aging heart. There's an increase in osteogenesis-related genes. If you want to look up these data, I think they're quite interesting and also confirmed. You can look in the publication. And there is a decrease in structural genes and also an increase in entropy and RNA velocity, meaning that the cells are more heterogeneous in the aging heart. Based on this first very strongly regulated angiogenesis gene in cluster 1, we phrased the hypothesis that maybe fibroblasts, by secreting angiogenic factors or by producing angiogenic factors, may interfere with endothelial cells and thereby contribute to endothelial cell aging in the heart. To test this hypothesis, we first used an in silico approach. This is a so-called ligand-receptor interaction, which was quite new at that time, and we did this in collaboration with Ralph Patrick, who wrote this program. And this is based on the understanding that if a cell expresses an agonist or a ligand, it could interact with a cell type B, which expresses a receptor, so either a secreted or a membrane-bound ligand, while a cell type C, which has neither agonist or receptor, couldn't interact. And this is counted in this in silico analysis, and we did also a weighted analysis, meaning what is regulated by aging. And it turned out that indeed fibroblasts and endothelial cells are strongly capable of communicating. So if you look in the total outgoing waves, meaning the signals produced, agonists produced by fibroblasts, there is the highest level here, while endothelial cells appear to have the highest number of incoming waves, meaning they are responding to the environment, suggesting that these two cells could indeed communicate. Further analysis then revealed that there are specific factors, and we ask whether aging fibroblasts by these factors may promote endothelial cell dysfunction in aging. As you know, aging is characterized by several hallmarks, and we investigated whether aged fibroblasts may affect endothelial cell functions. For this purpose, we isolated fibroblasts from young hearts or old hearts, then used the conditioned medium from the fibroblasts, added then to a matricial assay where we used endothelial cells, and then looked into the tube formation, either with young fibroblast medium or with old fibroblast medium. And you can appreciate here that there is a reduction in tube formation and also different other hormones of aging. For example, autophagy, which is preventing or maintaining endothelial health, was significantly reduced. Inflammation was augmented and sensitized here in the presence of TNF-alpha, while proliferation of endothelial cells was not affected, suggesting that old heart fibroblasts can indeed affect endothelial cells ex vivo. We did also analyze the capillary density in the regions of the heart and found out that you see here in the aging heart in this mice stream some patchy stainings for fibrosis. And particularly in this region, there was a reduction in capillary density, which suggests that fibrotic regions may be characterized by reduced either growth or even inducing capillary rarefaction in these fibrotic regions. Suggesting that cardiac fibroblasts may provide a paracrine niche in the aging heart, which reduces angiogenesis, reduces autophagy, increases inflammation, and thereby may contribute to endothelial cell dysfunction. The next question, of course, was what factors contribute to this? There were a number of factors regulated. I just show you here 4, but there were many, a large list of angiocrines factors and angiogenesis factors. And the top candidates were so-called SERPINs, SERPIN E1 and SERPIN E2. You know also these as PI1 and Nexin. They were enriched in these age-associated clusters, as you can see here. This is, again, this trajectory analysis. And we also confirmed this here in isolated fibroblasts of old hearts, which show higher expression of SERPIN E1. And we further also did now confirm this in the analysis of a published study. So this is a large sequencing data set of the adult human heart. And you can appreciate if we subdivide the females in more than 60-year and younger than 60-year-old individuals, there is an induction of SERPIN E1 and SERPIN E2. You see here, these dots all represent the expression level of these 2 factors, suggesting that's not only mouse heart, but maybe also in humans, there is an enrichment in these genes. SERPIN E1 and SERPIN E2, as said, PI1 and Nexin are maybe the better well-known names for it, are known to inhibit angiogenesis signaling. And we asked the question whether we can neutralize these SERPINs to prevent the inhibition of angiogenesis by the aged heart fibroblasts. To do so, we used neutralizing SERPIN antibodies, added this to the conditioned medium, and again used our tube formation assay. And you can appreciate that while in the aged fibroblasts tube formation assay, the conditioned medium here reduced again tubes, SERPIN antibodies prevented this reduction and led this to look almost like the young fibroblast conditioned medium tube formation assay in this context. You see here also the significant rescue, suggesting that SERPIN, although certainly not being the only ones, appear to play a very important role, at least in this ex vivo assay. This summarizes the findings. So we have shown that the aged heart comprises changes in cardiac fibroblasts. They release SERPINs. They can affect endothelial cell function. And interestingly enough, SERPIN E is also a marker for senescent fibroblasts or senescent cells in general. So it's known that this can be augmented in aged cells. And we asked next whether we might get rid of these cells by so-called senolytics and thereby prevent this negative crosstalk between old heart fibroblasts and endothelial cells. While the in vivo studies are still ongoing, we have some first evidence, but these are too preliminary. We have studies now in vitro, so we used senolytics. You see here, these are senolytics. They can be used to improve physical function and increase lifespan in mice. And one of these, quercetin here, it affects also SERPIN E and thereby can affect cells expressing SERPIN E. And we were questioning if we add this quercetin to the conditioned medium of old fibroblasts, whether we can rescue the function. And this was indeed the case. You see here the tube formation assay, old fibroblast conditioned medium reducing tubes, while the quercetin treated conditioned medium was much better. So the fibroblasts were much better. You can see here that these fibroblasts had no longer a negative impact, suggesting that maybe senescent fibroblasts may play a role here. This brings me to the conclusion of this first part of the presentation today. We know that there are many changes in the aged heart, and there are just many things I couldn't talk about, which are listed here. You can look into this review if you want, but certainly senescent cells play a particularly important role also, and we have now data showing, as I said, in vitro, that removal of senescent cells also can reverse the negative impact of fibroblasts on endothelial cells, at least in vitro. This brings me to the second part, which is a bit shorter and also unpublished. These are preliminary data. And here we thought, what happens in the human diseased heart? So most of the studies I have shown so far were in mouse, but the single cell sequencing also is usable in biopsies of patients. And therefore, we can disclose also cellular effects in the diseased heart. And to do so, we used the Human Healthy Heart Disease Atlas, which is published. I showed you this publication just before, with 14 healthy tissue samples. And we used hypertrophied hearts from our own department where we have patients with aortic stenosis which had hypertrophy. And then we performed a harmonization, which is quite complicated, but our bioinformatic groups really made sure that we could pool these data sets. And you see here the combined analysis of the healthy and hypertrophied heart samples. You see here the different clusters of the different cells. And you see also that both groups of samples are overlapping. So there is a nice harmonization. Then we asked, using this data set, similar to our age charts, what happens? How do cells communicate? And in this on the left-hand side, you see here now the cell communication in the healthy heart. This is the left circle, and the aortic stenosis patients. This is the right circle. And this is a circus plot now visualizing the communication pathways. Meaning, for example, in cardiomyocytes given in red, the red circle, the red lines represent the interaction with, for example, smooth muscle cells, pericytes, and so on. Here you see the fibroblast communication. And already, if you look at these circus plots, you see that in the healthy heart, there is a stronger line, so meaning more communications between the cells and cardiomyocytes. If you look in aortic stenosis patients, there is less communication, and also the lines are thinner, so the communication to the other cells by cardiomyocytes is reduced. We also counted the interactions, and you can see here there is a reduction of the interactions in the hypertrophied heart compared to the healthy heart, and also in the cardiomyocytes there is a particularly strong reduction in the signals. This would suggest that cardiomyocytes somehow lose the communication capacity, and we also quantified this. So this is the cardiomyocytes incoming signals. So you see here endothelial cells, aortic stenosis patients versus healthy. So cardiomyocytes communicate well in healthy, but less so in aortic stenosis patients. And this is the same for almost all cells. You see here always in the healthy heart, much better communication with cardiomyocytes. To give you some examples, one of the most strongly affected class of genes which were dysregulated and led to this reduction in communication were ephrin and ephrin receptors. These are pairs to be very involved in development, cell differentiation, tissue fibrosis, inflammation, and angiogenesis. We see here the ephrin receptors. There are two classes, A and B. There are many of them. And you can appreciate in the healthy hearts, each line here is one cell. You see here much more expression of ephrins compared to aortic stenosis patient, much lower expression. And particularly affected was ephrin B1 and A4, which are the highest expressed ephrin receptors in this data set. You can appreciate here ephrin B1 and A4 are the majority of ephrin receptors in cardiomyocytes, and both were dramatically reduced in response to aortic stenosis, suggesting that these receptors are silenced or downregulated on a transcriptional level in this context. For those of you who are not aware of ephrins, so ephrins are, as I said, a large class of genes which are comprising receptors, so the ephrin receptors, which I just have shown you, and then they can be activated by ephrin ligands, so ephrin A and B. You see here the ligands which can activate the receptors, and there's also complex signaling, so they can reverse signal to the cell which expresses the agonist, but also forward signal by activating the receptor. The ligand can activate the responding cell. These ephrin receptors make pairs with the ligands, as said here, and the two receptors which I've just shown you, which were downregulated in the diseased heart, were ephrin B1 and A4, which both can communicate with ephrin B2. This is the ligand for them. Ephrin B2 is known to be essential for cardiac development. Also, endothelial-specific deletion or blocking reverse signaling leads to development defects, so it's really essential. It's known to augment angiogenesis. It's pro-fibrotic, interestingly, so pro-angiogenic but pro-fibrotic, and it decreases the expression. It showed a decreased expression in cardiomyocytes during differentiation, so it has a very important function also on maturation of cardiomyocytes. So we thought that maybe the pair of ephrin receptor B1 and the ligand ephrin B2 may interact. And as you see here, this is the UMAP plot for cardiomyocytes, which showed the highest expression of this ephrin B1. And the ephrin B2 ligand was highest in endothelial cells. So this is an endothelial cluster here. The quantification is below here. So the receptor highest expressed in cardiomyocytes, the ligands highest expressed in endothelial cells. And interestingly, under disease states, the ligand is even further upregulated. So there is a upregulation of the ligand and at the same time, a downregulation of the receptor. So the question whether if you would block this FRMB2, which is the ligand for activating B1 at the FRMB1 receptor, would affect hypertrophy. And indeed, if we add this recombinant ligand, there is a reduction in hypertrophy, suggesting that this receptor ligand pair can prevent cardiac disease, at least cardiomyocyte response to phenylnephrine. In conclusion, these preliminary data would suggest that the crosstalk in the diseased heart is severely impaired. There is the expression of FRMB2, which is the ligand which is even upregulated in the diseased heart and which is strongly expressed by endothelial cells. While cardiomyocytes in the diseased heart, they reduce the expression of FRMB1, so they can no longer talk to each other, which may amplify cardiac hypertrophy and injury because this crosstalk is no longer existing. With this, I would like to conclude and thank the people who did the study, particularly Julian Wagner and our collaborators for the first part of the aging heart in the mouse. And also in the second part, Luca Nicin, Simone Glaser, and Sam Schröter, he's a new medical student who are working on this ephrine receptor interaction in the hypertrophied heart. And of course, all our colleagues in the clinic and our surgical department for their collaboration, and maybe particularly also David John and Ralf Schunze-Bühnig, who were very helpful in and essential actually in allowing us to gain these data and analyze these data also in these studies. Thank you very much for attention. I'm happy to discuss later on if there are any questions.
Thank you very much for this beautiful talk and showing so much data on animal and human in human models. And we will now go to the next step, and I will ask Professor Weber to introduce the next speaker. Christian.
Yeah, thank you, Tom. After a wonderful presentation by Stefanie, I would like to introduce Donato Santovito to the audience. Donato obtained his MD and also his PhD in experimental medicine at the University of Chieti, and he later on also obtained a habilitation in internal medicine in Italy. Then he moved to Germany, to Munich, to the Institute of Cardiovascular Prevention here at LMU Munich, and it was a pleasure to interact with him here as a postdoc and a senior postdoc, where he enriched also the collaborative research centers we have here in Munich on atherosclerosis and non-coding RNAs. Since last year, he also joined the Institute for Genetic and Biomedical Research at the National Research Council in Milan, and we hope to continue our interactions and collaborations. Donato has pioneered the non-canonical functions of microRNAs, in particular in endothelial cell biology, and also epitomized by a prototypic microRNA 126, which is important also in cardiovascular crosstalk, and also in these mechanisms that are subject to regulation. Autophagy plays a role, so there are some quite interesting links also to the presentation of Stefanie. And with this, I would like to look forward to your presentation.
Thank you very much. I share my screen. So thank you for the kind introduction, and thank you also for the great invitation and the opportunity to actually give me the possibility to show our data in such a high-level symposium. And in the next 15 minutes, we are basically speaking about microRNA 126 that is one of the most abundantly expressed microRNA in endothelial cells. There's several peculiarities, and one of them is the fact that both the guide and the passenger strand are actually functional and are potentially acting as a synergistic effectors of anti-atherosclerosis pathway. And in particular, while microRNA 126 3P is able to promote angiogenesis, plaque stabilization, and anti-inflammatory featuring by targeting the several pathways, including CXCL12 pathway. On the other way, I mean, microRNA 126 5P, that is the passenger strand, is able to preserve the endothelial cell proliferative reserve by targeting the DLK1 notch pathway. So now, in line with the different function, it's also known, we also note that the major strand of microRNA 126, namely 3P and 5P, undergo differential regulation in several stimuli that are important in atherosclerosis, but as well as in cardiovascular diseases we have seen in the previous talk. And this is in an example, the case of mTOR inhibition and autophagy. When we trigger indeed autophagy in endothelial cells by using rapamycin, we do see a strong reduction of the microRNA 126 3P, whereas the 5P was nearly unaffected. Both silencing of ITG5 and ITG7 that are required for the autophagy machinery for the vesicle enucleation and the elongation, as well as baphyllomycin, this actually required for blocking the lysosome and autophagosome interaction, thus preventing the degradative function of autophagy, were able to restore the level of microRNA 126 3P. But then we were wondering why the microRNA 126 5P was actually not affected. We hypothesized that the effect could be due to a different location. At the first, we transfected our cells, endothelial cells, with fluorescently labeled mimic for both 3P and 5P. And we did so that while microRNA 126 3P was localizing mainly in the cytoplasm and actually forming discrete foci upon treatment with rapamycin, on the other side, microRNA 126 5P show rather a nuclear localization that was strongly increased upon treatment with rapamycin. Obviously, this has been verified also for endogenous level by qPCR in a nuclear and cytoplasmic fraction. And we do see that the rapamycin treatment strongly increased the level of microRNA 126 5P in our cells, while the knockdown of ATG5, ATG7, or the treatment with ivermectin, that is an inhibitor of alpha-beta importing system in the nucleus, were actually producing an enrichment and an accumulation of the microRNA 126 5P into the cytoplasm. And together with microRNA 126 5P, actually we were able to identify that also EGO2, that is the main protein that interact with microRNA in mammalian cells, was also translocated into the nucleus. And the RNA immunoprecipitation confirmed the direct interaction preferentially with microRNA 126 5P. So now we were wondering, is this a specific effect for microRNA 126 5P or involve all the passenger strand of the microRNA? To this aim, we actually perform a small RNA-seq of nuclear or endothelial cells treated with or without rapamycin. We were actually quite also surprised to see that microRNA 126 5P somehow has the peculiarity to be the only one with a statistical significant enrichment in the nucleus following treatment with rapamycin. We perform mutational changes to understand what conferred this peculiarity to this microRNA. And interestingly, why the targeting the mutation in the seed sequence that is required for targeting messenger RNA did not affect the ability of microRNA 126 to shuttle into the nucleus. On the other side, mutations that are larger and go closer to the 3' of the microRNA were indeed able to disrupt this function. So we assume that this area should be somehow involved in this kind of selective process, while the targets are likely not to be involved. But what is driving then the 5P enrichment into the nucleus? As we saw that EGO2 was also translocated into the nucleus, we perform immunoprecipitation of EGO2 from both nuclear and cytoplasmic fraction of cells treated with rapamycin. And we screened for the expression, for the localization, of most of the proteins that are known to interact with EGO2, but that are also known to shuttle between nuclear and cytoplasm. And among that, we were able to find the RNA binding protein MEX3 as colocalizing in the immunoprecipitation, in the immunoprecipitated brought in the nucleus and cytoplasm with EGO2. These data were obviously confirmed by imaging, by confocal immunofluorescence, showing how by upon induction of autophagy by rapamycin, we found a nuclear localization of MEX3 EGO2 to form close localizing signal. We exploited in vitro by surface plasma resonance in immobilizing the EGO2 over a chip, the interaction between MEX3A and EGO2. It actually was quite surprising because EGO2 is not able to interact with MEX3A unless in the same system we were able to place microRNA 126-5P that somehow constitute the bridge between the two proteins. But what is MEX3A? MEX3A is a RNA binding protein and is a member of the MEX3 family that include 4 members, and is characterized by the presence of 2 KH domains that confer RNA binding properties, and a ring domain that is supposed to have E3 ubiquitinase activity. We use a molecular dynamic simulation model to analyze the potential way of interaction between microRNA 126-5P and the KH domain. And the best model was indeed a model where microRNA is able to bind both the KH domain, perhaps in a subsequential manner, perhaps with the KH2 before the KH1. This was confirmed by analyzing NMR spectra of interaction in presence or absence of microRNA 126-5P, showing clear shift in the localization of the residues. And moreover, we identified that concomitant incubation of KH2 domain with microRNA 126-5P was also able to produce a rearrangement of the domain toward a more stable, more compact, construct, as observed here by small angle X-ray scattering. This resulted in somehow a preferential enrichment and binding affinity of MEX3A toward microRNA 126-5P compared to microRNA 126-3P. That was determined in vitro by surface plasma resonance or isothermal titration calorimetry. And in vivo, in the cells, I mean, by immunoprecipitation of MEX3A that show a clear enrichment of microRNA 126-5P in endothelial cells treated with rapamycin. Finally, a functional role was also tested by knockdown experiment on MEX3A, showing that upon knockdown of MEX3A, we were able to prevent the rapamycin-induced nuclear enrichment of 5P, thus showing the relevant role of this protein in the shuttling of this microRNA. We then wonder if this nuclear localization actually reflects any functional relevance. To this aim, we first perform a transcriptional analysis of cells treated with or without rapamycin, with the first aim to put, obviously, microRNA 126-5P accumulating into the nucleus. This dataset has been then integrated with a second dataset where cells treated with rapamycin were treated also with inhibitor and mimic of microRNA 126-5P to promote further enrichment or complete depletion of microRNA 126-5P. By the integration of these two pathways, we were able to identify that the pathway linked to apoptosis were the most representative. And not surprisingly, by Western blot, we actually found that microRNA 126-5P transfection, together with rapamycin, were able to strongly decrease the cleavage of caspase-3, that is the main effect of caspase, and also decrease the cleavage of the substrate, like cleaved PARP, or also, if we want, cleavage of caspase-9. Obviously, these data were confirmed also by flow cytometry, and we do see that treating short-term cells with rapamycin already decreased the apoptotic rate in UVEC, and this effect is prevented by microRNA 126-5P inhibition, or even reverted by the knockdown of MEX3A. But what was quite surprisingly here was that this effect happened after 3 hours, while and well before the effect on gene expression are actually present. To this aim, we hypothesized that there should be a role of microRNA 126-5P somehow in regulating some fast mediator of apoptosis. And this fast mediator could be indeed an effect of caspase, as effect of caspase-3. At first, we tried to perform fluorescent in situ hybridization for microRNA 126-5P here in red, and immunostaining for caspase-free. And we actually saw already by confocal microscopy close localizing signal for these two components. Obviously, the nature of the findings prompt us for additional analysis, and we did also RNA immunoprecipitation. And surprisingly, immunoprecipitation of caspase-free from the nucleus of cells treated with rapamycin or exposed to high stress that we already shown to be involved in the promotion of autophagy in endothelial cells was associated with an increase of interaction with caspase-free, while in example, microRNA 126-3P was absolutely not interacting. This has been confirmed in vitro, again on surface plasmon resonance, on immobilized microRNA 126-5P and 3P, and we found that actually microRNA 126-5P was able to interact with the caspase-free, while the 3P was not. And this was finally also confirmed by NMR spectral changes that actually confirmed an interaction between the two components. And interestingly, we also found that MAX3 somehow favors this kind of interaction. Indeed, knocking down MAX3 in endothelial cells resulted in a lower amount of interaction between the caspase-free and microRNA 126-5P. But what is the relevance of that? We perform again molecular dynamic simulation to try to understand how the microRNA 126-5P can interact with caspase-free. At first, we were able to detect that the microRNA 126-5P was predicted to interact more likely with monomers rather than with dimers of caspase-free. And in particular, the interaction also involved this area that is the area of the heterodimer interaction. So we hypothesized that microRNA 126-5P could indeed disrupt the formation of the dimer of caspase-free. And again, by SPR, we were able to prove that increasing concentration of microRNA 126-5P were able to disrupt and prevent the further dimerization of caspase-free. So now we know that caspase-free dimerization is required for the formation of an active site. And so it was not surprising that in vitro, we found that addition of native sequence of microRNA 126-5P was able to dose-dependently reduce the ability of recombinant caspase to degrade its own substrate. This was the case for microRNA 126-5P, but not for the mutated 3, that is the one with the strongest mutation, and not, in example, for the microRNA 126-3P. But is this in vivo relevant? We actually previously showed that inhibition of autophagy in endothelial cells produce higher atherosclerosis in mice. Now we generate also another mouse model that is the mouse deficient of MAX3A. This has been generated by CRISPR-Cas9 model by placing a stop cassette at the end of the first exon. And we indeed saw that these mice actually show lower enrichment in the nucleus of microRNA 126-5P, in example, in the descending part, in the thoracic aorta. And this was associated with an increase with the active caspase staining, as well as by the detection of higher apoptotic rate by flow cytometry. And next, this could also have relevance in cardiovascular disease. Indeed, we found that in preliminary data, in preliminary experiments so far, that MAX3A mice in ApoE background also show an increase in atherosclerosis development. And our initial screening analysis also showed that some single nucleotide polymorphism in the MAX3A locus can be associated with cardiovascular disease in humans. But what we find pretty interesting is the inverse correlation between the eQTL for each one of these SNP and the odds ratio of association with cardiovascular disease. In example, for one particular SNP, we had that the disease allele associated SNP was associated with lower RNA expression for MAX3A, but still with an increase of cardiovascular disease. So, in conclusion, what we found is that autophagy can promote the nuclear translocation of microRNA 126.5p, and this preserves the level of 126.5p. But once in the nucleus, microRNA 126.5p require MAX3A to go into the nucleus and can dissociate from MAGO2 to bind caspase 3 and perhaps other caspases and prevent apoptosis. In conclusion, this is one of the first observations that microRNA can also act by directly modulating the protein function also outside of the RACE complex and potentially reveal new avenue for research and potentially therapeutic targeting in vascular disease and endothelial activity. And with this, I would like to congratulate, to thank all the people that supported our research in this year and all of you for your kind attention.
Well, thank you very much, Donato. And so basically we have heard very interesting presentations, and we can open a shared discussion with everyone. And Tom and I try to channel and funnel a little bit the questions that we've received here through the question and answer tool and direct them to Stefanie and Donato. So maybe I can just start, Stefanie. There are quite a lot of questions on your presentation, and maybe it was more relating to the first parts. People were interested to know whether there are also differential effects on different subsets on the crosstalk between the fibroblast and the endothelial cells, because you showed this endothelial cell heterogeneity, and basically whether different endothelial cell subset can respond differently, and maybe along these lines also whether this can also relate to regional differences between, for instance, the right and left ventricle.
This is a very good question. I think for SERPIN E1 and E2, we didn't find this to be enriched in specific subsets. So we couldn't define a cluster being, for example, only SERPIN positive, but it was rather, you have seen this also, it was more distributed. But this doesn't mean that there is not a spatial accumulation of such cells. And I think particularly the question of what happens in the left and right ventricle, I think so far has not been studied well. Now looking also in the comparison between right and left ventricle. And this could be also really interesting differential responses I can see in very preliminary studies. I also think that SERPIN E1 is not the only one. There might be specific subpopulation which we have missed, and we need these are just free mice. So maybe if you have a higher power and then select some cells, then maybe also you can get more insights into subsets.
Yeah. Great. And maybe, I mean, Tom, you kick in if you basically find something interesting. Also, some people were wondering whether you've put resident macrophages into the equation, you know, of this crosstalk, because they may also be picked up in your single cell analysis.
We didn't look into them. They are there. They are not so few. If this is not it's not an AMI, it's an aging heart. So the power is limited, and yeah, next project, I would say.
Okay.
Maybe partially related to that, a number of questions are actually related to potential heterogeneity and power of single cell analysis in human tissues. And there were questions about how to match the clinical samples related to age, potentially to clinical patterns, to severity of aortic stenosis, for example, or using different components of different cardiomyopathies. So could you comment on that in sort of a general term? And maybe it's a question to both of our speakers who also use human tissues. I also encourage our panelists to raise your hand if you have a question, and then participate in the discussion as well. So Stefanie, the heterogeneity.
So what I have shown now is, I would say, a relative large cohort, so 14 healthy humans, which were females and males, almost equally distributed, and also up higher than, older than 60 years, part of them, and 5 aortic stenosis patients, which are, yeah, relatively homogeneous. We have additional studied ischemic heart failure, DCM, and now also enrolled HFpEF. But we haven't now merged these data because already now it's such a large data set that we are having troubles to work with it. So I think it would be extremely interesting if we then can include more patients and collect data from different groups. And if we have good integration tools, this will be super interesting to look also for baseline characteristics and comorbidities, for example. Like if you have someone who has not well-treated diabetes, what does this affect the heart on a transcriptional level and things like this? I think we just started, and I think pooling data and sharing data will be very cool later on.
Yeah, and in particular, someone has also asked whether aging has been matched in these different pathologies that you're looking at, because that may also be an important driver of differences.
So I think the age was not, it's not entirely matched, but it's also not significant different because, as I said, we had samples from healthy humans older than 60 years, but they were a little bit younger. But we could also look in only those which were older, and the differences were very homogeneous in each patient and each healthy subject. So for example, ephrins, there was no age-driven relations. It was very significant, even if you do cluster T-tests and the normalization for the, and look only in those which are really age-matched.
Okay. And do you see specific differences between the pathologies or different signatures in terms of cardiomyocyte responsiveness? For instance, if you take your FNB2 model?
I haven't looked in the ephrin yet because these are really new data, but we see differences also in endothelial cells. For example, DCM and ICM appear to be very different. So dilated cardiomyopathy and ischemic cardiomyopathy, there's differences in inflammation, for example, in endothelial cells. But as I said, we have just started, and I will be curious to get more details.
So I also have a question with the FNB2, because as if I understood correctly, you see that basically a receptor downregulation on the one side, but then you get some rescue effect with adding FNB2 into the system. So how does this relate to potential also desensitization of the system on the receptor side?
So my idea is that the endothelial cell try to compensate for the reduction of FNB1. So they try to help in the end, but they can no longer reach because the signaling is down. So they can do whatever they can, but they don't respond anymore. Maybe because these ephrins can also signal reverse. So maybe if they do not bind, then this also gives the feedback to the endothelium, and somehow then this amplifies the response, but this is helpless. And the study in vitro, these were healthy cardiomyocytes. They were just PE treated, but protected previous. So it was not that we took a diseased cardiomyocyte and then added FNB2, but we did it before doing the stimulation. So it will be interesting to do more studies and get some more insights. What is hen and egg in this context?
We are joined today by Professor Karin Sipido, past editor-in-chief of Cardiovascular Research. Karin, you have a question, I can see.
Yes, thank you, Tom. I'll show myself. I wasn't planning to, but nice to invite me on the panel. Stefanie, many thanks for your talk. I thought it was really interesting. You know, we have quite an interest in fibroblasts as well. So my question to you relates to this concept of the aged fibroblast. Is that really a specific phenotype of a senescent fibroblast, or is it because of the environment in the aging heart, where you have inflammation, loss of aged myocytes? And do we know what the lifetime is of a fibroblast? What is the renewal rate? Because these are cells that have quite some proliferative capacity. So how does that compare to aging myocytes?
So it's a good question and also a research project. Maybe the short answer would be proliferation was not affected, at least according to a JCI Insight paper 2020 in aged fibroblasts. And it's hard for us to really dissect senescent fibroblasts. So we have difficulties to really understand whether this is true replicative senescence or whether this is more like a senescence associated phenotype, like having, certainly they have these senescence genes which are secreted. So these senescence proteins which are secreted, so these senescence factors. But whether this is a replicative senescence, we can't tell yet. We do more studies now to get more insights, but I can't answer this question.
But could you, for example,
senescence is not senescence, I feel that it's not the senescence phenotype with telomere shortening, but rather an activation and let's say inflamed pro-endocrine environment producing fibroblasts. This is not so different, but a lot of pro-inflammatory antigenic factors are changed. So this could be the phenotype.
Quite interesting. Thank you very much.
And kind of following on that, there is a question regarding your PhoneDB data, where you were showing the interactions between endothelial cells and cardiomyocytes. And it raised the question, what about interaction between fibroblast and an endothelial cell? What do you pick up pathways there?
Certainly, yes. They also changed. So this is very interesting as well, but they are not silenced. So this is more activated, so this communication. So maybe in half a year or so, I can tell you more about this. But it's different than cardiomyocyte, but also changed. So,
thank you.
Yeah, maybe an additional question from my end. I mean, in the SERPIN pathway of the fibroblasts, they also showed that the endothelial autophagy is one of the main effector functions that is affected and reduced on the endothelial side. So I was actually wondering whether, maybe also addressing this to the both of you, whether interfering or improving endothelial autophagy, which is also seen in Donato's data to be protective in atro, would also be a way of amending the aging phenotype in endothelial cells, or perhaps also using autophagy downstream pathways that are addressed then.
I maybe give this to Donato. So we have no data on it. I would predict it could work, but maybe Donato is the one who is more expert in autophagy than I am.
Yeah, I mean, I think we're very on the track to also analyze the role of autophagy in regulating also the senescence. I mean, the real problem perhaps in autophagy is to find a safe treatment that could be delivered and that could trigger autophagy without any side effect. So I hope perhaps some downstream effect of this autophagy pathway. I don't know, MAX3A could be the trick. But I mean, we'll take some months, years to tell you something more concrete.
Maybe I can have a question to both of you. It's kind of more philosophical question. It's related to some of the questions asked by our viewers, and a question that came to us on the Twitter. You should follow our journal on the Twitter if you don't. But the question relates to use of genetic data as well as RCT data, the clinical trial data, to substantiate and strengthen the basic science discovery. And you know, the main aspect is you know, when these data are supportive of your basic science discovery, then it's all great and wonderful. But what do you do if it's not? I mean, how do you deal with this sort of in inverted commas disappointment? But you know, we know very well that, for example, many targets of treatment of hypertension are, for example, not coming up in GWAS studies of high blood pressure. Yeah. Donato, maybe?
Yeah, I mean,
because you used the
genetic
data at the end, yeah?
That's a really tough question. I mean, was lucky enough to have some suggestion of involvement of MAX3A in our setting. I think it's a bit more complicated, and I mean, obviously, perhaps the best evidence is coming from either randomized clinical trial or by Mendelian randomization study to this end, or to data that will support like mediation analysis. As a matter of fact, it's not only matter of presence of mutation of expression of a gene, but as in the case, in example of MAX3A or autophagy, or for most of the marker of senescence, it's also the position or the localization and the activity of specific targets in the context of the specific cell environment. So on the other side, I mean, the bottom line would be, yeah, the genetic data can give us a good hint, but they are not conclusive in either way.
Any comments, Stefanie? You have huge experience in the human data.
Very interesting, of course, and maybe confirmatory, but you should keep in mind that only 1/3 of the patient's family history plays a role. So maybe genetics is not the only truth. It may be one aspect which is important, but there are certainly things beyond SNPs, which are important. Yeah.
Christian, anybody, any of the panelists?
So I think I've been asked
Stefanie?
my questions.
If I may ask a question for Donato?
Yeah, of course.
I was wondering whether you had a chance to look into caspase-1, because I was thinking that maybe your biological effects, if you would have an anti-inflammatory property of a microRNA, this of course may also explain some of the anti-arthrosclerotic effects. And
so yes, we started screening all the caspase, and caspase-1 for multiple reasons, despite being number one, it's among the target. We have to be a bit disappointing in this point, because apparently caspase-1 has not the same affinity for microRNA 126 compared to caspase-3. We actually identified perhaps 3 specific amino acids in caspase-3 that confer the higher affinity, yet are super preliminary data. At the moment, we are still developing the model for the molecular dynamic simulation, and we're trying to produce some additional data also with additional short sequence of RNA in vitro. But yeah, I mean, this would be really interesting to explore if microRNA or other short RNA actually can interact with anyone else of this caspase of other proteases.
And maybe the last point raised here by our participants is pharmacological targeting. Where do you think you can take these findings? And it's both related to Ephrin B and to Donato's work as well. Stefanie, regarding Ephrin.
There are possibilities to interfere with Ephrin. You can add Fc-linked recombinant ligands, and you can also use drugs which interfere with the pathways which are activated by Ephrin receptor-linked interactions. But I think it will be very complex, because, as I said, they have, for example, pro-angiogenic, but also pro-fibrotic activities. And to treat heart, I don't know, it's maybe not the best if you have fibrosis. And so there maybe we have to define which Ephrin has maybe specific functions and define targets which could be therapeutically used.
And for the microRNA, there are some more classical techniques, but have you identified any sort of maybe repurposing approaches, Donato?
Yeah, actually, in our setting, the idea to promote this non-canonical function would require both the activation of MAX3A and the presence of the microRNA in the right spot, in this case, in example, in the nucleus. We are working on the detail of understanding why MAX3A goes into the nucleus, if this involves, in example, changes in phosphorylation or specific nuclei localization signal. But yeah, we foresee as a complex approach to be pursued, but potentially, I mean, we require something more than simply delivering microRNA, if this is the final question.
Thank you very much. And if there are no other questions at the moment, of course, we cannot ask every question that was asked on the system. We will transfer these questions to our speakers, and they can answer to them through our journal or directly if such interaction would be valuable. Thank you very much on behalf of cardiovascular research to both of our speakers. We are very happy to have listened to such very interesting talks, and I will ask Christian to give some final remark. Thank you.
Yeah, Tom, thank you very much. And as usual, it was a pleasure to have this webinar, and we are also enlisted also in the ESC, worldwide webinars now. So basically, I think our audience can even further spread around, and we basically again have reached close to 400, 500 people in audience. And this is also thanks to our speakers and the really dedicated time and effort that they give to this, and producing really excellent and beautiful talks. So Tom and I thank you very much again, also for the really lively and vivid discussion, and most of all our two speakers, Stefanie and Donato. Thank you very much.
Thank you very much to everyone, and please join us for next seminars that we will have throughout this year. Thank you.